was investigating the role of GPR30 in inducing epithelial to mesenchymal transition in endometrial cancer. So my project mainly focused on endometrial cancer, and endometrial cancer is a type of uterine cancer specifically, and it's actually one of the fourth most common cancers in women. And as you can see here, uterine corpicus, which is endometrial cancer, in fact affects a large population of females. This specific cancer, in fact, is categorized into two main categories. So type 1, which is estrogen dependent, and estrogen dependent means that it should respond to estrogen and in fact should have an estrogen receptor. Whereas type 2 is classified as estrogen independent, and meaning that it doesn't respond to estrogen and shouldn't have an estrogen receptor. So looking at the progression of cancer specifically in endometrial cancer, and as you can see here, cancer cells start to begin to grow in the endometrium, which is the innermost layer of the uterus. And so as it progresses along, it starts to infect into other areas like the myometrium, and thus further along spreading into the fallopian tubes and the ovaries. And as you can see here in this case, it's actually infected into the myometrium. So cancer in general is considered to be the abnormal growth of cells. So as you can see here, the process of metastasis is mainly correlated with cancer in later stages, specifically the spreading of that tumor cell. As you can see here from this image, these tumor cells, if we think about them like back in the endometrium, this would be considered the endometrium, and this would be considered the myometrium. And this would be the basement membrane, kind of the barrier between the endometrium and the myometrium. And as that cancer progresses and those tumor cells grow, they start to infect into the myometrium and become invasive. And thus further along, they invade into the capillaries, progressing to other areas and infecting other parts of the body and organs and tissues. So kind of the main driver of metastasis is considered to be epithelial to mesenchymal transition. And what this means is that we originally start with epithelial cells, which are more stable, more structured, and less motile, as you can see here. And they are intact with their basement membrane. But as they progress and undergo that process of EMT, then they transition into mesenchymal cells, which break through that basement membrane barrier, and they become more motile and less structured overall. So EMT in cancer and metastasis, as you can see here, we start with that tumor that's very stable. Essentially, it would be epithelial, and then it would transition into EMT. It would undergo that process, thus invading into other areas and then entering into the blood vessels and traveling to other organs and tissues, and thus undergoing another process called MET, meaning that those mesenchymal cells that have been transitioned originally become epithelial cells, and thus locating in another area and colonizing and forming a tumor in another organ. So my project mainly focused on a couple different types of cancer cell lines. Don't know what that was, but okay. So I mainly focused on MCF7, which is a luminal A breast cancer cell line. A luminal A breast cancer cell line, and in fact this cancer cell line is estrogen dependent, meaning again that it does in fact respond to estrogen and should have that estrogen receptor. Whereas my other endometrial cancer cell lines that I decided to look at were RL952, which is a type 1 endometrial cancer cell line, and it is also estrogen dependent, meaning that it should respond in fact to estrogen. Whereas my two other types of endometrial cancer cell lines, which are type 2, are in fact estrogen independent. These are my HEC1A and my KLE. So that meaning that these two cell lines should not respond to estrogen and should not have that estrogen receptor present. And as you can see here from below, these are MCF7, my breast cancer cell line, that's very well categorized. Thus, that's why we decided to use it as a control. So main player in the process of EMT overall, we know, is considered to be estrogen signaling. And estrogen signaling can in fact activate a pathway called the PI3 kinase pathway, and that later on affects the process of EMT, the epithelial to mesenchymal transition. A main player in that estrogen signaling pathway, specifically in cancers, is known to be ER alpha. So this type of receptor, specifically in intracellular, meaning that the estrogen hormone can in fact enter into the membrane and attach to that receptor. So it's not membrane bound at all. And this receptor is involved in various types of regulation in gene expression and transcription. So thus we decided to examine the effects of estrogen on these cell lines that we wanted to examine. So what we did was count the cell lines and infect them with 1 nanomolar of estrogen. So our first cell line that we decided to look at was our MCF7, which we know to be estrogen dependent. And as you can see here, the red line is our monk, meaning that there's no estrogen present, and our blue line is our estrogen present. So as you can see here, there's definitely a significant amount of growth in the estrogen present cell line. And for our endometrial cancer cell line, RL952, which is also known to be estrogen dependent, you could also see that there definitely is a significant amount of growth for the cells that were exposed to estrogen. Whereas our HEC1A, which are our cell line, type 2 cell line, that in fact is considered to be estrogen independent, meaning it shouldn't respond to estrogen, we see that the red and blue lines overlap, meaning there was no significant growth difference between the two cells. However, for our KLE cell line, which is also considered to be just like HEC1A, a type 2 endometrial cancer cell line, and also estrogen independent, we do in fact see a growth, which we did not expect prior. We expected it to have no sudden growth and to be similar to the HEC1A. And from that, we stemmed our research question. So we decided to research how does estrogen signaling impact EMT in type 2 endometrial cancer cell lines, specifically that KLE cell line that we saw a significant difference in comparison to our HEC1A. So from that, we looked back into research and studies from other scientists, and we found out another receptor that could be used in estrogen signaling is GPR30. In this case, it's not intracellular, and in fact, it's membrane bound, and it's an orphan estrogen receptor. As you can see here, it's bound on the cell membrane in comparison to ER alpha, which is more intracellular. And both of them, in fact, can be involved in transcription. So from that, we decided to conduct a method called PCR, polymerase chain reaction. And what that does is basically amplify a specific gene sequence that we decided to look at. In this case, it would be GPR30 that we decided to look at. And so it has 3 main steps. Denaturation, meaning that that DNA strand that we're looking at is being broken down. And then our second step, which is annealing, meaning that the primers that are specific to our GPR30 are attaching to the site that we need. And extension, meaning that continuous amplification of that sequence that we need to create multiple copies. And then we decided to analyze it via gel. So as you can see here, we're looking at our GPR30, and in our MCF7 cell line, which is our positive control, we can see that GPR30 is in fact present. And this cell line is estrogen dependent, meaning it should respond to estrogen. Whereas our HEC1A, which is our endometrial cancer cell line, and we know to be estrogen independent, it does not showcase any GPR30. However, for our RL952, which is a type 1 endometrial cancer cell line, and it is also estrogen dependent, just like the MCF7, we can also see that there is a band for GPR30 present there. However, for the case of KLE that we saw prior, and we wanted to examine, we do in fact see a faint band present for the GPR30, meaning that GPR30 is present in that cell line. And then we decided to use actin just as a control to make sure that that data that we received from our GPR30 gel is in fact correct. So some main take-homes from this so far is that our KLE cells express GPR30, and they do in fact respond to estrogen, on the contrary of what we had known prior. Also, our HEC1A, which is our type 2 endometrial cancer cell line, and is known to be estrogen independent, meaning doesn't respond to estrogen, showcases that it doesn't respond to estrogen and lacks both the GPR30 pathway receptor and the ER alpha receptor. So as for our MCF7, which is our positive control, we see that there's both expression of GPR30 and our ER alpha, and in fact it does respond to estrogen. So from that, we decided to explore more about GPR30 and looking specifically at what would be GPR expression, and the presence of an inhibitor for that GPR30 pathway in relation to understanding how does estrogen travel through GPR30 and how does that factor into the pathway. So from that, we decided to do an assay called immunofluorescence, and what this assay basically looks at is the specific membrane protein that we would like to examine, in this case GPR30 again. And so for this assay, we have our antigen, which is our protein that we want to look at specifically, and we add a primary antibody first, which attaches, and thus we add a secondary antibody, and that secondary antibody has a fluorescence probe on it, which allows us to see it, which allows the cells to fluoresce if the protein is present in the cell line. So from that, we decided to examine it in the presence of estrogen and G15. So our mock is with the lack of GPR30 and the G15 inhibitor. So not our GPR30, but our lack of estrogen present in that sample. So as you can see here for our MCF7, there is in fact the presence of GPR30 there, entailed in the, if you can see the red kind of dot surrounding that, and the blue is the nuclei present. And you can see the same here for our KLE cell line. So as for when we treat them with estrogen, we definitely see a significant increase in protein expression for GPR30. As you can see, there's definitely a lot more red surrounding these nuclei. And as for our last part, which is both treating them with estrogen and G15, the inhibitor specifically for GPR30, we can definitely tell that there's a decrease in the amount of GPR30 expression present here, similar to, again, to the mock trial, which there was no estrogen present and no inhibitor present in that. Whereas for our KLE, you can also tell there was a significant decrease from the estrogen. However, it's not similar to the mock, and we decided to conclude that may be due to the specific inhibitor that we used, since we had retrieved it from previous studies that mainly targeted MCF7 rather than the KLE cell line, which may have caused some non-specific binding for our cells. So from that, we decided to explore the mesenchymal marker. And basically what we were looking at for that specific process is to understand further whether or not GPR30 and its estrogen response has anything to do with the EMT pathway, that epithelial to mesenchymal transition, which mainly relates to metastasis again. And so what we decided to look at was vimentin, which is our mesenchymal marker. And from that, you can see in our mock, vimentin is in fact present for both mock and G15 and estrogen. However, when being treated with both that G15, our inhibitor, and our estrogen, which is stimulating these cells, you can definitely tell there's a significant decrease in the vimentin marker, that mesenchymal marker that we're examining. Actin was just used to determine whether or not our data again was conclusive and whether or not what we had retrieved from our data was correct. So the main conclusion that we came to at the end was that KLE cells in fact do lack the ER alpha, but that doesn't mean that they don't respond to estrogen. They do. Whereas our KLE cells in fact do express GPR30, and that might be, again, the relationship into why it responds to estrogen. So we also saw from the previous data that the inhibition of GPR30 specifically decreases that mesenchymal marker that we were examining, vimentin, meaning that it might have some sort of relationship with the regulation of EMT. Thus coming to our final conclusion that GPR30 may be the main estrogen signaling pathway in KLE cells, or type 2 endometrial cancer cell line, and that might be factor into the regulation of the process of EMT, that epithelial to mesenchymal transition, which would allow us further along to understand whether or not that may impact metastasis and so forth. So some future directions that we came to was that to further examine our GPR30 protein expression in our endometrial cancer cell lines, and we decided that we could try another assay, specifically called Western blot, and that also examines proteins, but more so on a gel rather than fluorescing the cells. Another thing was to examine additional epithelial and mesenchymal markers, and that could be expressed, so to help us understand further along that relationship to EMT in terms of the regulation of GPR30. And some of those markers could include, for example, ECadherin for epithelial markers, and then L1CAM for decided that we need to conduct another immunofluorescence assay, just because our prior assay did show that there might be some sort of non-specific binding that might have been caused to our inhibitor that we had used prior to that. And we wanted to determine whether or not those data that we had received prior to that was in fact conclusive or not. So I wanted to acknowledge a couple of people. Dr. Irene Reed for being a great faculty mentor and supporting me throughout my research, as well as the USJ Biology Department for allowing me a space to conduct my research in, and the USJ Provost Student Research Award, as well as the Beta Beta Research Award grant that allowed me to fund my research, as well as my amazing lab mate, Gina Galucci. Thank you.